This procedure begins by removing any debris from the Influenza A virus solution by centrifugation. The viral membrane is then disrupted with a detergent, and the released VRNPs are separated on a glycerol gradient centrifugation. Fractions from the gradient are collected, and an aliquot of each fraction is run on an SDS page. The gel is stained with Kumasi blue to determine the peak fractions containing VRNPs. Fractions containing the VRNPs are then concentrated by centrifugation, and the purified VRNPs are applied to a copper TEM specimen grid, which is then negatively stained for visualization of the VRNPs by transmission electron microscopy.
Hi, I'm Winko Wu in the laboratory of Dr. Nellie Pante in the Department of Zoology at the University of British Columbia.
I'm Lindsay Weaver, formerly from the Pante Lab.
Today we will show you a procedure for the purification of Influenza A viral ribonucleoprotein complexes.
We use this procedure in our laboratory to study the nuclear import of Influenza A virus in tissue culture cells.
So let's get started.
To begin the experiment, add 750 microliters of MNT buffer into one 11mm by 34mm Beckmann polycarbonate centrifuge tube. Next, add 500 microliters of a 2 mg per milliliter solution of Influenza A virus into the tube. Pipette up and down several times to mix the virus with the MNT buffer. After mixing, place the tube in a TLA 120.2 rotor and centrifuge in a Beckmann Optima Max E centrifuge for 10 minutes at 109,000 times G at 4 degrees Celsius. When the spin is done, remove the supernatant. Then resuspend the pellet in 500 microliters disruption buffer. Vortex the resuspended pellet vigorously, and then shake at 31 degrees Celsius for 20 minutes in an Eppendorf thermomixer. When the shaking is done, proceed with the glycerol gradient sediment velocity centrifugation. To prepare the glycerol gradient, place the following amounts of glycerol into a Beckmann Ultra Clear 13mm by 51mm centrifuge tube. 1 ml 70% volume by volume glycerol, 0.75 ml 50% glycerol, 0.375 ml 40% glycerol, and 1.8 ml 33% glycerol. These glycerol solutions are made by mixing pure glycerol with NM buffer. Also prepare a balance tube containing both glycerol and buffer. Then vortex the disrupted viral sample again and load it onto the glycerol gradient. Place the tube in a Beckmann MLS 50 swinging bucket rotor and centrifuge the gradient for 3 hours and 45 minutes at 217,000 times G at 4 degrees Celsius. After the centrifugation is complete, manually collect 250 microliter aliquots of the gradient starting from the top of the tube. Keep fractions on ice or at 4 degrees Celsius. Once the fractions are collected, we are ready to analyze them using SDS polyacrylamide gel electrophoresis. To begin the SDS page analysis, remove 20 microliters from each fraction to a fresh tube. Then add 5 microliters of 5 times SDS page sample buffer and heat the sample to 95 degrees Celsius for 5 minutes. After heating, spin down the samples briefly and load them onto a 10% polyacrylamide gel. Include molecular weight markers in one of the wells. Next, run the samples through the gel until the bromphenol blue loading dye reaches close to the bottom of the gel. Remove the gel from the SDS page apparatus and stain the gel with Kumasi blue. Based on the staining results, Identify the fractions that contain mainly Influenza A nucleoprotein. The Influenza A NP is approximately 56 kilodaltons in size and is the major protein found in viral ribonucleoprotein complexes. So the NP band generally is the strongest band in the fractions containing the VRNPs. In the next part, we will concentrate these VRNP fractions. Combine the glycerol fractions containing mainly NP and distribute them into two Beckmann 11mm by 34mm polycarbonate centrifuge tubes. Fill each tube with diethyl pyrocarbonate or DEPC treated ultra-pure water. Then pipette up and down several times to mix. Place the tubes in a Beckmann TLA 120.2 rotor and centrifuge for 4.5 hours at 157,000 times G at 4 degrees Celsius. When the spin is complete, remove the supernatant and resuspend the pellet in 50 microliters of DEPC treated water. Aliquot the purified VRNPs. Set aside one aliquot and store the others at -80 degrees Celsius. Now we are ready to proceed with the negative staining of VRNPs. Dilute 1 microliter of purified VRNP into 9 microliters of freshly made and twice filtered PBS buffer. Glow discharge a copper TEM specimen grid for 30 seconds. This grid was previously coated with a Parlow dion and carbon film. Using tweezers to hold the freshly glow discharged specimen grid, apply a 5 microliter drop of diluted VRNP onto the specimen grid. Allow the drop to absorb onto the grid for 8 minutes. While waiting, place a small strip of parafilm in a tray. Then dispense onto the parafilm 2 drops containing 10 microliters each of PBS buffer, as well as an 80 microliter drop of the freshly made staining solution, which is composed of 1% ammonium molybdate. Now wash the specimen grid by carefully lowering it into the 2 drops of PBS for a total time of 1 minute. Then use a piece of filter paper to wick off part of the sample solution from the specimen grid, without letting the specimen grid dry. Immediately after wicking off the last drop of buffer from the specimen grid, completely submerge the specimen grid within the stain droplet and wait 1 minute. After 1 minute, use a new piece of filter paper to completely wick off the stain solution from the specimen grid. Allow the specimen grid to air dry for several minutes before observation under a transmission electron microscope. Here are representative transmission electron microscopy images of negatively stained VRNPs. As you can see here, the VRNPs resemble rod-shaped particles with variable length that are approximately 30 nanometers to 120 nanometers in length. The oligomeric NP is organized as a chain of NP molecules that is further folded into a double helical repeat structure, so loops on either ends of these rod-shaped particles can sometimes be seen.
We've just shown you how to purify and visualize Influenza A viral ribonucleoprotein complexes.
When doing this procedure, it's important not to use PBS when diluting the VRNPs if you're using uranyl acetate for negative staining.
So that's it. Thanks for watching and good luck with your experiments.